Hejsan och varmt välkomna till Schizofrenidagen. Jag heter Emma Frans och jag kommer vara er moderator i eftermiddag. Jag är andra dagar forskare här vid Karolinska Institutet och vetenskapsjournalist. Jag skriver bland annat för Svenska Dagbladet. Det är idag första gången Schizofrenidagen arrangeras här vid Karolinska Institutet. Och jag blev väldigt glad när jag fick frågan om jag ville medverka här idag. För jag tycker verkligen att den här dagen har ett viktigt syfte. Och syftet är att vi vill berätta om pågående forskning och nya forskningsresultat om psykisk sjukdom med fokus på schizofreni. Vi vill att fler ska få ta del av hur kunskapsläget ser ut idag och vilken typ av forskning som bedrivs. Därför riktar vi oss idag till alla som är intresserade av att veta mer om psykossjukdomar, allmänheten, patienter och anhöriga, vårdpersonal och andra forskare. Det talas allt mer om psykisk ohälsa och stigmat kring dessa diagnoser minskar successivt. Och detta är en väldigt positiv utveckling. Men ofta så ligger fokus på depression eller kanske ADHD. Och schizofreni och andra diagnoser får inte riktigt den uppmärksamhet som det förtjänar. För schizofreni är ett vanligt tillstånd som drabbar upp till 1% av befolkningen. Och dess allvarlighetsgrad varierar, men för många så innebär tillståndet stor funktionsförlust under lång tid. Så idag kommer vi uppmärksamma schizofreni och ni kommer få höra populärvetenskapliga föreläsningar från personer som forskar om psykossjukdomar. Ni kommer till exempel få höra hur man kan göra nervceller från en bit hud och vilken betydelse den här tekniken har för forskning om psykisk ohälsa. Ni kommer också få höra om spännande forskning som hittat kopplingar mellan psykisk ohälsa och kreativitet. Vi kommer också ha en fikapaus där ni har möjlighet att ta del av en utställning som finns här i foajén med information om Svenska Schizofrenistudien, psykossjukdom och Riksförbundet Hjärnkoll. Ni kommer att ha möjlighet att ställa frågor idag efter varje föreläsning. Och vi har också avsatt ordentligt med tid på slutet av dagen med en timmes frågestund. Och tanken är att både ge er en möjlighet att ställa frågor inför oss allihopa. Men också att ni ska kunna ställa enskilda frågor till varje föreläsare via ett mingel. Och alla föreläsare kommer att vara med i slutet av dagen. Och ni kan ställa era frågor, om ni har papper så kan ni skriva ner dem på lappar. Så vi kommer ha behållare som samlar in de här lapparna och det kommer också finnas lappar tillgängliga. Och ni kan också ställa frågor direkt via mikrofon. Det finns också möjligheter att ställa frågor via Twitter. Vi har en hashtag, Schizofrenidagen är den. Och ni kan också mejla in frågor. Så innan vi sätter igång så ska ni bara få några korta säkerhetsföreskrifter. Vid en eventuell utrymning så kommer ni höra ett talat utrymningslarm. Om ni hör det så följ de instruktionerna som ni får och ta er ut via närmaste utrymningsväg som är markerat med en grön skylt. Här och här. Och det finns också utrymningsledare på plats med gul väst för att visa er till återsamlingsplats vid en eventuell utrymning. Så då skulle jag vilja börja med att presentera vår första föreläsare. Det är professor Patrick Sullivan som är professor och psykiatriker vid institutionen för medicinsk epidemiologi och biostatistik här vid Karolinska Institutet. Patrick Sullivan är också professor vid University of North Carolina. Han leder genetiska studier för schizofreni, depression, ätstörning och autism. Han har även startat det största samarbetsprojektet någonsin inom psykiatri, Psychiatric Genomic Consortium, där över 800 forskare arbetar tillsammans. Han har publicerat flera hundra forskningsartiklar och det är Patricks forskargrupp på Karolinska Institutet som har arrangerat Schizofrenidagen. Varsågod.
So, Emma, thank you so much for the introduction. And thank you all for coming today. I apologize for my poor Swedish. I will try to speak slowly in English with translations alongside. I'm going to begin with thanks. So the work that we've been doing here in Sweden, going back to 2003, 2004, has had many, many people who have made incredibly important contributions to all of it. The list of people here is partial. There are many other people who have contributed in small and very large ways. Most of them are here. In particular, I would like to thank Anna Kaler, who is the person who actually organized much of the work, much of this conference today. In addition, Christina Hultman has been my longtime colleague, Christina Dahlman, Håkan Karlsson, as well as many other people within Sweden who have very kindly worked with us over the years. But in particular, I'd like to acknowledge the incredibly important contribution of the Swedish Research Council and funders in the United States as well as in the EU. So the thing I'd like to begin with, just to make sure we're all sort of on the same page, at least partially, is, and these are sort of the key points I'd like to make today. The first thing is that schizophrenia, I think, as everyone in this room knows, is very complicated. And we certainly do not want to minimize the complexity and how important it is for an individualized approach to this. But at the same time, we've known for some time that genetic studies actually make a lot of sense to do. The rationale, the logic behind doing them is definite and clear. And increasingly, we're making very real progress. I'll talk about this in a little bit, but genetic approaches to trying to understand schizophrenia are really starting to work in important ways. In addition, I think many people may not be aware, but Sweden actually has been at the center of a lot of this work. A lot of what we know about schizophrenia comes from Sweden. A lot of the work goes back to things that Christina Dahlman and Christina Hultman and others have been doing for many years by studying Sweden as a large population in terms of trying to understand what are the risk factors for schizophrenia. But more recently, the genetic data we've been doing have been based in large part on Swedish samples. We've certainly done a lot, but I believe that we do need to do more. The thing that's kind of fun is that as the genetics is progressing, a lot of other scientists are becoming involved. And so many of us have multiple active collaborations with people in neuroscience who are working hard to take the genetic findings and try to understand what's happening at a very detailed biological and even a cell-based level. So the thing that the question mark I put here at the end is, are we in fact entering into a golden age of schizophrenia research? I certainly think so. But people have accused me for many years of being overly optimistic about everything. So you have to take that as, with a grain of salt. Okay, so some beginning comments. So to go over some of the basics. So the definition of schizophrenia requires at least 6 months of active symptoms. It's not just having a bad day or a bad week or something strange that happens. It really has to go on for a long time. And classically, the definition, at least in part, is due to the presence of persistent delusions, which are fixed false beliefs, and/or hallucinations. In other words, false sensory perceptions. And again, this requires a relatively intense amount of symptoms over time. Again, it's not a simple thing. It's not a very brief thing. But we also know from other work, again, a lot of it coming from Sweden, is that schizophrenia is really among the top 10 conditions in the world in terms of its impact on people. Technically, they talk about the days of disability-adjusted life years lost. Effectively, big phrase for the fact that schizophrenia really messes up a lot of people's lives in some very important ways. In terms of mortality, in terms of death, the life expectancy of people in Sweden is some 10 or 15 years less than everybody else, which certainly indicates its massive impact on health. Obviously, I think the impact on a personal level, on a family level is huge. If you want to put a number on it in terms of cost, The amount is something in the order of 500,000 Swedish crowns per person per year in terms of direct and indirect costs of schizophrenia. And that's from a Swedish study that was done a few years ago. We also computed a couple years ago that if you think about extended families, around one family in every 79 in Sweden is impacted by schizophrenia in one way, shape, or form. Now, sorry, I'm doing things. So I wanted to talk about what's the goal? What are we really trying to do? And typically we talk about sort of two basic ideas for what genetics is trying to accomplish. Really, the first step, if we can, at some point in the future, is prevention. Can we actually find a way to identify people who are at high risk and then do something before they actually develop schizophrenia to keep it from happening in the first place? Can we prevent it from occurring? That's a long-term goal. The other thing that, of course, we're trying to do is to try and develop better treatments for schizophrenia. As many people who have had anything to do with somebody with schizophrenia certainly knows, the treatments we have can be effective for some people at some times. But most of the treatments are not curative. They prevent, they minimize symptoms. But they're not where I think anybody wants them to be in terms of dealing with the full impact of schizophrenia on people. We need improved treatments. And to do both of these correctly, to eventually figure out how to prevent, as well as to develop better treatments for people, we really do need to understand the biology deeply. So if we think about cancer, for instance, some areas of cancer, the treatment is radically different today than it would have been 5 years ago. For instance, the treatment of melanoma, like advanced melanoma, life expectancy has gone from something on the order of 5% to like 40 or 50% now, because a deep understanding of the biology of melanoma has allowed people to develop better treatments by precisely focusing on the exact parts that go wrong in the creation of this. And I would argue that for schizophrenia, the same thing holds. We have to understand what it really is biologically, what really is going wrong in great depth in order to prevent and in order to treat better. And next question is, how do we actually go about doing this? What I'm showing here is a map of a consortium called the Psychiatric Genomics Consortium. The PGC has been in business for around 10 years. We're the largest consortium in the history of psychiatry. There's currently over 800 scientists actually in the PGC, and I think there's around 1 million genetic information from around 1 million people actually in the PGC right now. As you can see, the countries in blue are the ones where we have active participation, and as you can see, we actually have most of Europe, a lot of Asia, North America, big parts of South America, obviously less in India, as well as Africa, but there's efforts ongoing right now to improve that. But one of the reasons why this is working is that we've been able to bring people together effectively. This truly is an international effort, and the reason we're making progress is because psychiatrists and scientists who work on this are working together every day in what I think is a very effective way. So why genetics? And it's relatively easy to state the problem, and I've mentioned this before, is that if we really wanted to do rational therapeutics, if we want to develop the right treatment for the right people, we really do need to understand biology. Historically, this has been very difficult in psychiatry. In fact, most of the medicines that we have that are being used right now in psychiatry were discovered by accident. The history of the first antipsychotic medicine was that a chemist in the early 1950s was trying to develop a better antihistamine. The person apparently wasn't a very good chemist and actually made the first medicine, which is an antipsychotic. And all subsequent antipsychotics have been not designed rationally, but by making small changes on this, et cetera. It's been really more of an opportunistic process rather than a design process, and that's something that we need to do. In addition, I think it's certainly been the case that every generation has really tried hard to solve schizophrenia. So if you go back and read the literature from 100 years ago, people tried hard using whatever technology they had at the time to make a difference to schizophrenia, to make a difference in trying to fix the basic problem. And it's been very difficult, and that's because schizophrenia is tough. And I would argue that one of the few things that we have that have actually worked are genetic approaches. And it's kind of interesting, but you can actually determine that this is a hard problem, but it's not an impossible problem. The universe of solutions for what's going on with schizophrenia is not infinite. There are 20,000 genes in the human genome, and something like 12 or 13,000 of them are expressed in the human brain, which means that that is not an infinite problem, but actually it is something which we can gain traction on. And obviously the key thing with genetics is we're going to use genetics to help us identify what are the important genes, and that will, we hope, will be the thing that will actually provide the foundation for the work that we need to do. And in terms of just why genetics, this is a slide that goes back a ways in terms of how old it is. But basically, it's a major clue. And this goes back to observations that people have been made for at least 70 years. But the thing that's interesting about it is that it's really sort of a probabilistic, not a deterministic sort of genetics. Genetics does not doom a person to develop schizophrenia. It increases the risk. When people think about genetics, often they think about a genetic variant which, if it's absent, the risk of a disease is 0, and if it's present, the risk of disease is 100%. That's not the case for schizophrenia. All the things that we have are subtle. They increase risk, but they never get it to 100%. In fact, the largest genetic risk factor that we know of right now increases risk only up to around 20%. And so it's the case, even if you have that particular rare genetic variant, most people with it will not get schizophrenia. So that's kind of a key point, that really this is something where the risk factor is really more subtle than it is absolute. And in fact, we know that something on the order of only 5 or 10% of people with schizophrenia will have an extended family member with the disease. If you think about monozygotic or identical twins, if one twin has schizophrenia, the risk to the co-twin, who is genetically identical for most intents and purposes, is only on the order of 50%. And so again, I would argue this means that this is a subtle process. The slide I'm showing here lists a number of risk factors for schizophrenia, all the way from polio infection to brain damage at birth, German measles, and other things that damage the CNS. And this is by how much risk is increased. And so, for example, if there's marked damage of the brain at birth, the risk goes up around 7-fold. If you've got a family history of schizophrenia, the increase in risk is on the order of 10%. But again, that means that if a person has, if there's a person with schizophrenia in a family, on average, the risk will go from around 1% to around 10%. The vast majority of people who have a family member with schizophrenia will not themselves get schizophrenia. And wanted to go over a few things about genetics real fast. Again, I have 2 slides. This will be brief. Many of these things are a much, much longer conversation. But the basic idea is that we all inherit, every cell in our body, almost all the cells in our body have on the order of 3 billion bases. That's a 3 followed by 9 zeros. And these are the things we inherit. We get 1 copy of most chromosomes from our mother, 1 copy from our father. There are other types of changes can occur, but by and large, most of what we get is determined here. And these are the things that make us up, for why we have certain hair color, why we have certain height. Very few of these things are directly determined. There's always a role for environment, for what we're exposed to, for what we choose to do, for what happens to us, etc. And there's a level of complexity as well, which is actually an entire another lecture. But the 2 things I'm going to focus on today are there are some changes that occur at a single base level. One of these letters that make up the book of DNA with 3 billion letters, 1 letter might be changed. Some of these are inherited, Some of these are mistakes, errors that happened in the making of us. The second type is there's a level of change that occurs called a copy number variant. Usually we get 2 copies of genes, one from mom, one from dad. There are certain regions where we'll only have 1 copy, or we'll have several copies. And these are regions that they occur for a particular reason in terms of what they don't happen by accident, they happen because of a logic that occurs in the genome. But a few of these have been strongly associated with schizophrenia, with autism and other things. And I'll summarize those in a bit, but that's the second type I'm going to talk about today. So things that change single bases as well as things that change many bases at a time. Okay, so one of the things, so part of what we do is this cartoon sort of illustrates what happens for one of these single variants. This is a situation where if we look at a group of people who we call a case, people with schizophrenia, for instance, versus people without schizophrenia, controls, we might find that everybody's got 2 of these variants, a C or a T in terms of the particular letters that are at one very specific spot in the genome, but we might find that the frequency is 62% for this one, but only around 50% for that one. This is the kind of signal we're looking for, something where we can look at one of these and find a place in the genome that's really quite different in people with schizophrenia versus people that don't have schizophrenia. And it's possible now with our technologies to do this a 500,000 times. So instead of doing just one of these, we can do many, many of them that are scattered approximately evenly throughout the genome. And when we do this kind of work, and if we do it carefully, we can then circle back and see, okay, this is a huge difference. We can't make it go away. We don't think it's a mistake. We think it's a real finding. Then what gene is it in? And what does that gene, what we know about what it does, how does that help us understand schizophrenia? That's the basic logic of the work that we do. And I'm going to walk through sort of the time scale. I'm going to go from 2005 up to the present time. And going back about 10 years, honestly, we only had about 1 strong or confirmed risk factor for schizophrenia, just 1. Then there was a paper in 2008 where it went from 1 to 5. Next major paper was in 2009 that brought it up a bit more. And then the next one up got us to a total of 11. And then this one is a paper that came mostly from Sweden, actually, and that took the total up to about 30. That was 4 years ago. We published a paper 2.5 years ago where the number went from 30 up to 120. And then there's an unpublished paper right now where our colleagues have identified 150 different places in the genome that are involved in schizophrenia. And in about a month, the consortium that I mentioned before is going to have an analysis of 71,000 people with schizophrenia, which is about twice the number that we had in 2014. So as you can see, this is what my contention for why things are moving quickly. We've had this massive explosion in knowledge about the genetic basis of schizophrenia. And we're pushing it really hard. It's got a lot of focused attention. There's a lot of people in the world that are working on this, and we're quite confident and hopeful that we'll actually learn a lot more in the near future. Okay, so let me just sort of summarize where we are right now. The first is, what's our estimate of the number of genes when we're finished with this work that are going to be involved in schizophrenia? My guess is it'll be in the high hundreds. So not quite 1,000, somewhere between 700, 900 genes we will determine have a role in schizophrenia. Next question is, how common are the genetic changes that we're talking about? The vast majority of the ones we've discovered so far are common and have very soft, very subtle effect. In fact, they're so common that most of us in this room have inherited quite a few. And so this is one of the things that I mentioned before, is that it's a subtle thing. The genetic basis of schizophrenia increases the probability of the disorder, but it rarely, if ever, means you will get it. So it's probabilistic, not deterministic. Next, the sizes of the genes that the effects have for are mostly small. There are a handful that are somewhat larger. We know that most of these just act in a very simple way. There's a bunch of complicated ways that genes and environment can act or interact. But honestly, we haven't found much evidence of that. It appears that the basis of schizophrenia appears to be relatively straightforward. And this is sort of a graph of the summary of what we found so far. On the top, these pie charts show intellectual disability, autism, and schizophrenia. And the colored areas refer to the strong genetic findings of a couple of different types. Won't go into the details now, but for intellectual disability, for instance, if you do a full genetic workup, you can probably find an answer for severe intellectual disability for maybe 2/3 of kids with this condition. For autism, the similar number is around 1/4. For schizophrenia, it's only a couple percent, probably 2 or 3%. But the point here is that I would argue this kind of makes sense in a way, that if you've got a condition which is very severe and very early onset, that's one thing. But for schizophrenia, where many people develop, mostly okay, usually, it makes sense that it's really more of a subtle process, and that's typically what we see. There's certainly exceptions to what I just said, but I think as a first approximation rule, it's probably okay. So again, the earlier onset, the more likely to have a very strong variant. The later the onset, perhaps less likely. This shows a graph of how common things are. So this is 1 in 10, 1 in 100, 1 in 1,000, et cetera. And this shows by how much it increases risk. So 2 means it doubles the risk of schizophrenia. 3, 10, 10-fold risk, and the highest one here is up at 80. So as you can see, there are some things that have been discovered which are uncommon or rare, but have a relatively strong effect. The ones in red are ones that were determined from looking at changes in the numbers of copies of genes, and there's only one that's really pinpointed one particular gene. But the vast majority of it is actually coming from out here. These are things that I'm showing about 160 different dots there, and these are things that are present in 1 out of 10, and yet have a very soft effect. And that's where most of the signal's coming from. And when we look at the common things that we found, it's not a random set. They tend to focus in certain sets of genes. So for example, genes that are involved in how calcium works in our brain, how it signals things in the brain, are actually relatively strongly enriched. There's also a couple of very specific biological processes that have come up. As it turns out, there's ways in which the nerve cells in our brains are changed or remodeled, and that appears to have something to do with schizophrenia as well as several other types of biology that appear to be involved. Okay, so what can we do in Sweden? Now I'm going to start by thanking Christina again, who was involved with the Broad study going back to 2004. I think I took Christina and Paul Lichtenstein out for a Thai meal near St. Eriksplan and sort of convinced her over beer that this was worth doing. I hope she thinks that it was a good thing to do, because I certainly do. And these are some of the papers that have come up that have been based upon the sample that Christina directed and collected here in Stockholm. And it was a national sample, but it's been extremely informative. And the study that we're talking about we call S3, which is the Sweden Schizophrenia Study. And in the prior version of this, Christina and colleagues managed to identify 5,500 people with schizophrenia, 6,500 controls. At the time, that was the largest sample in the world. There's now a larger sample. And as a consequence, or because we need to learn more, we're in the process right now of trying to add in more subjects. Everything we do is subject to informed consent and full explanations of everything. We only want people involved who really want to be involved. And we currently have collected something like 1,300 people with schizophrenia and 5,000 controls above and beyond these other ones. And we're in the process of doing more. If I could point to Anna, who is right there. And Isabel is where? Isabel, there's over there. These are the two people who are on the front line right now in collecting and getting more people into the study. If you or someone you know would like to get more information, please let us know at the break. We'd be happy to help. In addition, there was the email address that you put up at the start, and that's another good place to contact as well. Okay, so the next thing is that, again, this really is evolved into a major international effort. There's a lot of scientists involved. I know of at least 3 Nobel laureates whose labs are working on schizophrenia right now, and they weren't 10 years ago. And a lot of this is because of the genetic side. What we're trying to do really, really hard right now, we're pretty much all out on this, is to try and learn more about the biology of schizophrenia and then to see if we can circle that back to become clinically relevant. And effectively what we want to do is we want to take the family history risk factor and convert that into something which means something for people. This is not just something that academics do because we like learning things. We want it to matter. We want it to come back and make a difference for people in Sweden. This is a paper that came out last year where we were able to pull together a conference where we had a lot of people from industry and academics to really come together and try and think through how the two efforts could actually help each other. And this sort of summarizes really briefly the work that I do that's relevant here. So as Emma mentioned, I'm half in Sweden, half in the States. And if I'm a little bit fuzzy, I apologize. I was in the States in summer a few days ago. And I'm kind of jet-lagged beyond belief, but I'm doing my best. But what we have here is just basically the major projects in my group. So this is the Sweden schizophrenia study, which is we're working on hugely. This is a study called GAPS, which is basically trying to actually do genomics in all of Sweden. We've managed to get a lot of people interested in this. This is less for schizophrenia and more just for general precision medicine. We have an active project called SUN, where we're actually working with several neuroscience labs to try and figure out what the implications of the genetic findings are. We have a study called COSIN, where this is an EU-funded project where we're trying to essentially make cells from specific patients to see whether we can find a treatment that works for that person. And Anna Falk will be talking about this later. And then we also have a lot of work in the States where we're doing a lot of ways to try and understand what's going on in particular brain cells as well. And just to sort of talk about technology a little bit, and this is some of the stuff that has sort of really engineered or made this possible. This stuff is, the science behind this is crazy. I don't understand a lot of it. We know it works, and the engineering behind this is actually wondrous. But this is sort of a way that we can actually, for any person in this room, we could do 500,000 genetic markers in a couple of days. And the cost is about $30 US. The first time we did this about 10 years ago, the cost was around $1,500. So the cost has come down hugely. This is the technology that we use to resequence somebody, to basically take DNA from anyone and actually go back and look at pretty much every base all over again to see what they have that might be different than anybody else. The first time this was done, the cost was in the hundreds of millions of dollars, and now we can get it done at SciLife Lab down the street for about $1,000. And this is a technology that's coming online now. It plugs into a USB drive on your computer. You put DNA in, and it does the sequencing using just the power from the USB drive. So this stuff, there's a lot of people working on the technology. The stuff that's possible now is just incredibly cool, and a lot more is happening as well. Really, really briefly, this is some of the technology that some of my neuroscience colleagues can do. So this is, for example, they can look at nests of neurons at scale and do it automatically, measure 40 different things per thing. They can increase the amount of a particular gene, decrease the amount, put a medicine on to see what happens, et cetera. And all of this can occur in more or less real time. Another colleague of mine can actually look at neurons really directly. So we can take a neuron from a human and grow it, oops, sorry, the slide didn't advance. And we can take a neuron from a human, put it on one of these little dots. This is a picture of a neuron. All the little green things is where the neuron touches itself, a synapse. And he can do tons of stuff, like they can image individual synapses. They can take movies to watch them over time. He can put a needle in right to that spot and measure exactly what's happening at that synapse. I mean, this is the degree of resolution that we're going to need in order to make things, to make this work. Mentioned very briefly, this is a longer talk. I'm working with Jens Erling-Leffler here at KI, and they can actually do studies in individual cells. So they can take individual brain cells and interrogate them in incredible depth and resolution. And that technology is something that we're combining with the genomics that we're doing to take it further. Okay, so predicting the future is hard, but I'm going to hazard a prediction. I think that we finally have the right toolkit for us to really make a very strong push toward understanding the biology of schizophrenia. And I think actually it's quite possible that we're entering into a golden age for schizophrenia research. We'll see. Maybe I'm right, maybe I'm wrong, but I'm totally all in. We've got a great lab, a lot of great colleagues, and we're pushing this as hard as we can. And our goal is, as soon as we can, to try and start doing some things that make a real difference for the lives of people with this often devastating illness. Okay, that's it for me. Thank you very much for your attention.